So we move on with the third chapter that we have, and that's enzymes. Okay. So does anyone here know what enzymes are? How would we classify? Let's say if you would have to describe enzymes in two words, how would you describe enzymes as? So whenever we would want to describe enzymes, we would say that enzymes are what they are biological catalysts, right? Now, before we move ahead with the definition, let's talk about what catalysts are. देखो catalyst कोई भी ऐसा substance होता है, catalyst is any substance that would speed up the rate of reaction. Now, during this time, it's not going to be consumed in any way. It's going to, stay how it was initially. The only thing that it's doing is that it's enhancing the reaction, or it's, let's say, making the reaction occur at a faster pace. Now, if I'm saying that the rate of reaction is increasing and in the presence of catalysts, it means that the reaction of whatever is happening is taking less time to occur, right? Now, we can have catalysts outside living organisms as well. One of the things that we'll study in your chemistry is that the transition elements, they are catalysts. What are transition elements? Iron, nickel, copper, vanadium. You can talk about them in chemistry. You'll talk about iron in the Haber's process. You'll talk about vanadium in the contact process. You'll talk about nickel in organic chemistry, so on and so forth. Now, what we need to let's separate out is that those in catalyst, or they're since they're inorganic catalysts, they're not going to work inside living organisms. Enzymes are the catalysts that will work in the organisms, right? So for hence we are saying that हमारे पास जो enzymes हैं, they're biological catalyst. कि भाई they are going to work in living organisms. They're, let's say, they would be found in this things that are living, right? Whether it's animals such as humans, whether it's plants, whether it's bacteria, whether it's fungi. So you're going to have enzymes everywhere. Uh, it would not be incorrect if I would say that there is no biological reaction that could take place in the absence of enzymes. सही है, हमें enzymes चाहिए होते हैं. देखो बच्चे, what are enzymes, Hiba? Enzymes are those catalysts, those substance. So again, what is a catalyst? If I'm saying catalyst, this definition goes for every catalyst that there is. Okay, a catalyst would speed up the rate of reaction. It would make the reaction occur faster without getting used up. Uh, being biological means that they would be found only in living organisms. All right. So hence we say कि भाई अच्छा ये enzymes हमारे पास जो हैं ये क्या हैं? They are biological catalysts. Those catalysts which will be found in living organisms only. Now, what are they made up of? हम कहते हैं भाई enzymes are biological catalysts. What in nature? Made up of what? हम लोगों ने कहा कि भाई they are protein in nature. They are made up of protein. Now, what are they doing? So the rest of the definition is just what we just explained earlier, that enzymes are biological catalysts made up of proteins or protein in nature that speed up the rate of reaction, right? But the real question is how. तो हम लोगों ने कहा कि भाई the enzymes, they are biological catalysts that are going to increase the rate of reaction. But the thing is how? कैसे? किस तरीके से? Right? हम कहते हैं कि भाई enzymes or whatever the catalyst. So again, this is true for all catalysts. All catalysts work in the same manner. So whether enzymes, whether inorganic catalysts, they speed up the rate of reaction. How? By lowering the activation energy, या फिर energy of activation. So we're going to underline this word. So enzymes, along with all the other catalysts that we have in the world, What do they do? They speed up the rate of reaction. How? By lowering down the activation energy. Now, in this topic, before we're going to talk about how the enzymes work, how many types of enzymes do we have, what are the factors influencing the rate of enzymes. सबसे पहले हम लोग बात थोड़ी सी activation energy के बारे में करेंगे. So does anyone here know what activation energy is? क्या होती है activation energy?
The minimum energy required to start a
Very
reaction.
good. Very good. Very good. So activation energy is the minimum amount of energy needed to start a reaction, right? Acha, now if I were to explain this to a 5-year-old, what am I going to do now? देखो, होता क्या है? Any reaction that we have, any chemical reaction, before the chemical reaction could occur, it needs a small investment. ठीक है? It needs a small bit of investment. It needs some sort of energy to start. That energy that the reaction needs to start, no matter how small, no matter how great. There are some chemical reactions which require a lot of activation energy. There are some chemical reactions which require very less of it, right? But the thing is that initial energy that is needed to start up a reaction is what we call as activation energy. Up again, what example can we take from a daily life? If I would go into the kitchen and if I would turn on the stove, what's going to happen is there's going to be gas that's going to be issued from it. It's going to be methane gas. Methane gas reacts with oxygen in the atmosphere to produce carbon dioxide, water, and a lot of heat. So let's say if I start, or let's say if I would turn on the stove and do nothing. So I have a stove from which that methane gas is coming out. It's reacting, or it's, the atmospheric oxygen is also present. Will that stove catch fire? So what I only did was I just turned on the stove. I just allowed that gas to, to come out of it. I already have oxygen that I'm breathing in the atmosphere. So if I'm just going to turn on the stove, is that stove going to catch fire? A simple yes or no question, guys. It will need energy to start the reaction. ठीक है. Hiba, I haven't applied anything yet. I haven't applied, let's say, I haven't applied a spark. I haven't applied any sort of flame to it. I just went and I just turned on the gas, nothing else. The stove would not catch fire. It would keep on issuing the methane gas, but it's not going to light up. ठीक है, आग नहीं लगेगी उसके अंदर. When, as Uzair said, it's only going to catch fire when you're going to provide it with a small amount of energy. Now that energy could be a spark, right? So we usually most of our stoves that we have nowadays, they come with a built-in spark, right? So you just press on the knob and it will start going tick 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 tick. Or अगर हम लोग, if you go back 10 years, we used to light a fire. We used to light a matchstick so that that particular gas that's coming out of the stove could catch fire. Up that energy that I'm applying so that these two reactants, the methane gas and the oxygen, they could start a chemical reaction. तो देखो chemical reaction होगा, right? लेकिन उसको शुरू करने के लिए मुझे क्या चाहिए होती है? मुझे activation energy चाहिए होती है. अब that energy could be anything. हमारे example के अंदर it was electrical energy in form of a spark. It could have been heat in form of a matchstick. Let's talk about another process. We have a process that occurs inside the plants called as photosynthesis, right? Photosynthesis को शुरू करने के लिए हमें क्या चाहिए होता है? Light चाहिए होती है, and lots of it. So what is the purpose of light in photosynthesis? Is acting as an activation energy, right? अब enzymes और catalysts क्या कर रहे हैं? They are just decreasing the activation energy. कि देखो, अगर if that investment is there, or if the activation energy is there, And if it's, let's say, if it's high, right, if it's great, what will the enzymes and all the other catalysts do is उसको कम कर देंगे कि अच्छा ये रिएक्शन देर से शुरू होना था क्योंकि इसको एक्टिवेशन एनर्जी कलेक्ट करने में टाइम लग जाएगा, हम क्या करते हैं, लेट्स डिक्रीज इट डाउन सो दैट दिस रिएक्शन कुड स्टार्ट ऑफ विद लेस एनर्जी. Ariba, no. Uh, and chlorophyll, yes. Chlorophyll, Uzair, is the substance in which photosynthesis is occurring. Chlorophyll is trapping light. Light किसकी तरह काम करी, वो activation energy की तरह काम कर रही है. अच्छा, so Ariba, instead of spark is not functioning, we press, हाँ, you don't press on the knob, just, turn it down. You don't even have to press it. So press कर दो, the spark comes out. If you don't press it, and if you just turn it on without the spark, right, the gas would keep on coming. And again, until and unless you're provided with heat, if you would provided with the right amount of activation energy, it would not catch fire. Right? Acha. Khair. Acha, let's take another example. What's basically happening? Let's say I go all Willy Wonka and I want to start, or I want to open up a chocolate factory. Now let's assume कि भाई, to open up a chocolate factory, I require $100,000, for example. ठीक है? Now let's say if I'm saving $1,000 per month, how long is it going to take me to, reach this amount? It's going to take me 100 months, right? Acha, Abdullah ne kya kara? Abdullah said, how about I give you $50,000? आपके पास हो दे दीजिएगा, नहीं हो मत दीजिएगा, right? And you can like use this to open up the chocolate factory. Now I have half of the investment with me. To reach $100,000 again, if I'm saving $1,000 per month, now instead of 100 months, how much time will I require to reach the very same amount? I would require 50 months. Why? Because what I'm receiving is an initial investment. You can, Abdullah, you can have chocolates every day. Forget every month. All right. So what actually happened? The final amount that I had to reach, did it change? It did not. What only happened was to reach this amount, I now required less time, or let's say in our example, half the time, just because I received an initial investment. Ab ek reaction hoga, it has to happen, right? It will start, but it's going to take a longer time to start. What are the enzymes going to do? They're going to start the reaction early because that reaction now would need less energy to start. अगर एक चीज जल्दी शुरू हो रही है वो जल्दी खत्म भी हो जाएगी, right? Let's put it this way. Let's put it this way. We'll just do two more examples and then easy समझ में आ जाएगी. How do enzymes, or when anything that works with the enzyme, sleep early and wake up early, exactly. Acha. So let's say the things that react, or the substances which usually we call as reactants, enzymes के अंदर उनका नाम बदल जाता है. The reactants that we have that interact with the enzymes are called as substrates. Substrates, in the presence of enzymes, are converted into products. Let's take an example. Starch is a carbohydrate usually found in bread. It reacts with water molecules, so it could convert into another sugar, smaller sugar, called as maltose, right? So let's say I put a piece of starch in my mouth. The water is there. Let's say 2 mole per dm cube of starch will take 4 hours to be converted into maltose. But in the presence of the enzyme amylase, the very same reaction is going to occur in 4 seconds instead of 4 hours. देखो हो क्या रहा है, हो क्या रहा था, starch and water to react to be and starch to be converted to maltose, it requires a lot of energy. My mouth has some sort of heat energy, but that is too less. It's too less. So the starch molecules and water molecules, if they're present there, they're going to collect that energy, collect that energy. But it's going to take a very long time for them to collect that, or to reach the activation energy that they need. The enzyme amylase comes along and says, okay, you require 100 joules of energy. Okay, how about you start the reaction in just 1 joule of energy, right? I'm going to provide you with an alternative pathway so that you are able to start the reaction, but you'll just you know consume, or you would require less activation energy. अब इसको इस तरीके से देखो, इस तरीके से देखो, whatever the reactants that we have, or the substrates in our case that we have, they also have a particular amount of energy, right? Now these substrates to be converted into products, they require this much activation energy. So EA is activation energy. So activation energy can also be written as EA, energy of activation. And finally, producing what? Products, which would also have some sort of energy. Now, this is the time from this point towards this point. This is the time taken for the reaction to be completed. अब एक enzyme आया. What did this enzyme do? I'm just going to copy this down so it's easier for us. So what is this enzyme going to do? It's the very same reaction, right? This enzyme is just going to decrease the activation energy. Now, when the activation energy is decreasing, the time taken to complete the reaction has also decreased, right? पहले मुझे इतना सारा टाइम चाहिए था, अब मुझे कितना टाइम चाहिए? I just require only this much time. And we said it in the last chapter कि भाई किसी भी चीज का जो रेट होता है ना, work done over time, अगर टाइम कम हो गया हमारे पास रेट ज्यादा हो जाएगा. Now all in all, if you would see here, nothing happened to the energy of the substrates that they possessed. Nothing happened to the energy of the products. It's still the same. I literally copied the same image. I just decreased the activation energy. So enzymes or the other catalysts that don't alter anything except for the activation energy. Now, this is something that we have to keep in mind, not only for biology, but also for chemistry. You're going to study enzymes and rates of reactions. यहाँ तक बात समझ में आ रही है बच्चों? यहाँ तक कोई मसला? Any questions? Does anyone want me to repeat this again?
Sir,
जी मेरा बच्चा.
sir, you mean that if there are enzymes, so like any reaction can be done in less time?
In less time. Yes.
Okay. Thank you.
That's it. That's the gist of it. ठीक है. आबरू बच्चे कोई मसला?
No, thank you.
ओके. अच्छा जी. अब देखो, अब हम लोग ना इसको थोड़ा सा केमिस्ट्री के व्यू पॉइंट पे पढ़ते हैं. Why? Because we have some MCQs that revolve around this. There are these things that are called as energy time graphs, right? What do these energy time graphs tell us? ये बताते क्या हमें, right? हम बताते हैं energy time graphs tell us two things. Number one, the overall energy change in the reaction. कि अच्छा अगर substrate और products के बीच में कोई change आया है, so how much energy was given out and how much energy was taken in, that this is something that is not our domain. वो chemistry वालों का है, exothermic endothermic energy. ठीक है, बस हम calculate करना सीखेंगे. The second thing that they tell us is the activation energy, right? अच्छा, ओवरऑल एनर्जी चेंज का हमारे पास सिंबल होता है, इट्स अ ट्राइंगल विद एन एच, एंथैल्पी चेंज डेल्टा एच। एक्टिवेशन एनर्जी के लिए जो हम लोग सिंबल लेते हैं, वी गोइंग टू टेक इट एज ई ए, राइट? सो दिस सिंबल्स, ताकि मैं दोबारा लिखूं तो मुझे पूरा नहीं लिखना पड़े। अब हाउ or what would that energy time graph look like? A typical energy time graph has energy on the y-axis in kilojoules or joules, and we have time in on the y-axis of it's seconds, minutes. We are not concerned about the time. What we're concerned about is the energy. Up, let's say by mere pass 20 kilojoules, 40, then we have 60, 80. Let's take it all the way towards 100. Up in a reaction, I had a substrate that had energy of 60 kilojoules. I had products that had energy of 20 kilojoules, right? Now, how would that graph look like? Now, this graph would look something like this. I'm sorry, just give me a minute. Okay, right? अब इस ग्राफ से मुझे दो चीजें कैसे पता लगी? सबसे पहली चीज, the very first thing that I have to find out is the overall energy change, right? How do we calculate the overall energy change? Energy of the substrates minus the energy of the products, right? So what was the energy of the substrates? 60 kilojoules. What was the energy of the products? 20 kilojoules in our case, right? So you can be given a different graph. Concept ये apply होगा बच्चों. Overall energy कितना use up हुआ या release हुआ इस experiment के अंदर? 40 kilojoules, right? फिलहाल इतना पता होना चाहिए कि overall energy हमारे पास कैसे निकल, ठीक है? पहली बात ये हो गई. Now for the thing that we need, activation energy. ठीक है? अच्छा, अगर हम लोग उसको ग्राफ पे देख लें, अगर फॉर्मूला याद नहीं रहे ना, कुछ नहीं करिएगा, सबस्ट्रेट और प्रोडक्ट्स के बीच के अंदर एक लाइन बना लीजिएगा. So you mark the energy of the products, you mark the energy of the substrates, and you draw an arrow like this. So it's going to give you the answer right away. ठीक है? Now for the activation energy. Activation energy is calculated by subtracting the energy of the substrate from the highest energy reached minus energy of the substrates. Up in our case, highest energy reached कितना है? Let's take it as a 90. Energy of the substrates कितना है हमारे पास? 60. तो activation energy कितनी इस्तेमाल हुई? 30 kilojoules. कहने का मकसद क्या हुआ कि भाई, to start this reaction, you first need to give it an energy worth or equal to 30 kilojoules. भाई फिर ये substrate या ये reactants products में convert होंगे, वरना नहीं होंगे. अब what do enzymes or the other catalysts do? वो क्या करते हैं? वो इसी activation energy को कम कर देते हैं. So this was the highest energy reached in absence of enzymes. This is the highest energy reached in the presence of an enzyme. Up now, if I were to calculate the activation energy in the presence of the enzyme, अब enzyme ने क्या करा? Highest energy reached को reduce कर दिया, right? वो 90 से कितना ले गया उसको? Let's assume this to be a 70. Substrate की energy कितनी है? वही 60. अब in the presence of enzymes, अब activation energy कितनी लगानी पड़ी? 10 kilojoules. So देखो, पहले हमारे पास क्या हो रहा था, अगर हम लोग देखें, this is how you found the activation energy, right? In the presence of the enzyme, the activation energy has been decreased. Now, to start up this reaction, I don't need 30 kilojoules of energy. उसको थोड़ी सी 10 kilojoules of energy हम लोग provide करेंगे, वो reaction शुरू हो जाएगा. Right? So this is how enzymes work. यहाँ तक कोई परेशानी? Any questions at all? जी मेरा बच्चा. Sure, 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 sure. You want to copy this down? अब Zeb? हाँ, sure. Copy कर लो. अच्छा जी. So now that we know what our activation energy is, अब हम लोग बात करते हैं enzymes के बारे में. ठीक है, enzymes how do they look like? How many types of enzymes do we have? ठीक है, इसको देखते हैं. अच्छा, सबसे पहली बात कि again, I'm just going to talk about that equation once again. कि भाई we have molecules that react with enzymes called as substrates. Substrates, right? By the action of the enzymes, they are converted into products faster. Now, how does an enzyme look like? So if you would talk about the structure of an enzyme, all enzymes look nearly identical, right? Except for one part. So this can be an enzyme, this molecule that I have drawn right here, this could be an enzyme. So enzymes are pretty much the same, except for these indentations, these gaps that they have. These indentations are called as active sites, right? So what are these? This is an active site. This is also an active site. An active site is the working part of the enzyme. Enzyme kya karega? It will allow the substrate molecules to come to its active sites, where it will allow the activation energy to be decreased so that the reactants could or the substrate could be converted into products. Now, enzymes are doing what? They're helping or they're decreasing the activation energy or they're speeding up the biological chemical reactions. We have a term that we give to biological chemical reactions. Any chemical reaction that is occurring inside your body is what we call as metabolism. It's a collective term. Metabolism क्या हम कहते हैं ना भाई, this person has a very fast metabolism, this person has a very low metabolism. Metabolism is just a technical word that we give to all chemical reactions that are occurring inside the body. Digestion के अंदर भी हम लोग बात करेंगे, hydrolysis होता है, that's a chemical reaction. Things join up, condensation, that's a chemical reaction. Respiration, that's a chemical reaction. Photosynthesis, that's a chemical reaction. Transmission, generation, nervous impulses also involve chemical reactions. So all of this is a part of your metabolism. The food that I'm consuming is being broken down. The nutrients from the food, they're being chemically removed, or the waste products are being chemically removed. Urea formation होती है metabolism में, right? जब भी हम लोग मेटाबॉलिज्म के बारे में बात करते हैं ना, तो मेटाबॉलिज्म कंसिस्ट ब्रॉडली, हम कहते हैं भाई मेटाबॉलिज्म में से दो चीजें हो रही हैं. या तो चीजें बन रही हैं या चीजें टूट रही हैं. Either substances are being built, either substances are being broken down. I'll give you an example. Those chemical reactions, no, no, chewing is not a chemical reaction. Why? केमिकल चेंज नहीं हो रहा ना, फिजिकल चेंज हो रहा है. फूड टूट रहा है, फिजिकल उसमें चेंज आ रहा है, केमिकल चेंज कोई नहीं आ रहा. दैट इज नॉट, दैट्स अ फिजिकल ब्रेकडाउन, नॉट अ केमिकल ब्रेकडाउन. दैट्स नॉट डाइजेशन. खैर, हमारे पास या तो एनाबॉलिक रिएक्शंस होंगे या फिर हमारे पास कैटाबॉलिक रिएक्शंस होंगे. एनाबॉलिक रिएक्शंस वो वाले रिएक्शन जब चीजें जुड़ती हैं. कैटाबॉलिक रिएक्शंस वो रिएक्शन जब चीजें टूटती हैं. Right? Let me give you an example. I like steaks. Steaks are a very good source of proteins. I had a steak. I had that, let's say that piece of meat. And once, so it's basically, it has a high content of protein, right? Proteins are molecules that are too large to pass from the digestive system into the blood. So what happens is my digestive system breaks it down, digests it, hydrolyzes it. In other words, it's breaking it down into smaller molecules called as amino acids. Now, those amino acids, they move into my blood, and let's say at the same time I'm working out. Okay, I'm hitting the gym every day. Now, those amino acids, when they will go to my muscles, they will be joined back up to make the proteins that I need. Some of those amino acids may go into my salivary glands and produce the enzyme amylase, right? So अब हो क्या रहा है? When the proteins were being broken down into amino acids, that was a catabolic reaction. चीजें टू प्रोटीन टूट रहा है, वही अमाइनो एसिड्स मैंने लेकर क्या करा, अपने प्रोटीन्स बना लिए, उनको दोबारा ज्वाइन अप करा. Anabolic reaction. It's just like playing with Legos, right? So I had a Lego की कार, मैंने उस कार में सारे लेगो निकाले और उन्हीं लेगो के पीसेस का मैंने क्या करा, मैंने एक हाउस में छोटा सा घर बना दिया. Are we getting this? Now, इसी तरीके से we have two types of enzymes only. So if we talk about the types of enzymes, we have two types. The first type can be a catabolic enzyme. Another type is an anabolic enzyme, right? So enzymes that break the substrates to give you products, catabolic. Enzymes that joins up the substrates to give you products, anabolic. So let's draw these. So let's draw these. So let's say the catabolic enzyme pretty much looks something like this. The anabolic enzyme, let's say, looks something like this. अच्छा, अब जो मेरे पास catabolic enzyme को जो substrate है ना, that is like this. That substrate, when it binds, only when it binds onto the active side. देखो बच्चों, enzyme substrate reaction की पहली condition क्या है? The substrate should bind onto the active side. We're going to write that down. लेकिन पहली condition क्या है? The substrate is going to bind to the active side. Once it does, the substrate is converted into products, right? In the case of the catabolic enzyme, the substrate will be broken down into the products, and those products will then leave the active site, right? In case of anabolic enzymes, the substrates would go onto the active sites where they will be joined to form the product. And once the product is formed, it would leave the active site. Do we get this? यहाँ तक बात समझ में आ रही है बच्चों की भाई how this whole thing is working out?
Sir, can you please explain catabolic again?
देखो, catabolic or catabolism is whenever substances are broken down. Those enzymes that carry out catabolic reactions where they have to break down the substrates into products, right? They will be called as catabolic enzymes. And the opposite is anabolism or anabolic enzymes.
So why these enzymes leaves active sites?
No, enzymes are not leaving the active sites. Active site is a part on the enzyme where the substrates are binding. अब मेरे पास substrate product में convert हो गया, right? अब substrate product में convert हो गया. That product will now leave the active site. I'll tell you the reason why. ठीक है, वरना ये वाली बात समझ में आई? डिड वी गेट दिस स्ट्रेट के सबस्ट्रेट एक्टिव साइट को लीव नहीं कर रहा, सबस्ट्रेट एक्टिव साइट पे जाके लग रहा है, एंजाइम फिर उस सबस्ट्रेट को प्रोडक्ट्स में कन्वर्ट कर रहा है, एंड देन द प्रोडक्ट्स आर लीविंग द एक्टिव साइट।
Yes, sir. I understand. Thank you.
ठीक है, यू वेलकम। अच्छा, अब मुझे एक छोटी सी बात ये बताओ, will the substrates of the anabolic enzyme, can they bind to the active site of the catabolic enzyme? मेरे पास जो anabolic enzyme का substrate है, वो catabolic enzyme के ऊपर जाके लग सकता है? नहीं रख सकता। वेरी गुड। सिमिलरली जो मेरे पास ये कैटाबॉलिक एंजाइम का सबस्ट्रेट है, कैन इट बाइंड ऑन टू द एक्टिव साइट ऑफ द एनाबॉलिक एंजाइम? नहीं, ना, नाडा। ठीक है, क्यों? शेप अलग है ना, लेकिन शेप सेम नहीं है। इस समय दो बातें पता लगी ना। सबसे पहली बात हमें क्या पता लगी, मेरे पास एंजाइम्स जो होते हैं ना, दे आर स्पेसिफिक इन नेचर, वी हैव अ स्पेसिफिक एंजाइम फॉर अ स्पेसिफिक सबस्ट्रेट। हम कहते हैं वो, एंड व्हाट बेसिकली let's say does this is the specific shape of the substrate. हम कहते हैं the substrate, shape of the substrate should be complementary. Now look at the word complementary, right? Should be complementary to that of the active side. अब complementary का हरगिज़ मतलब ये नहीं होता कि भाई same हो। Complementary का मतलब क्या होता है? Things that go together, like raita on biryani, fries and ketchup on fries, right? So fries and ketchup go together, raita and biryani go together. Chocolate and biryani would not. They don't complement each other. Right? Do we get this? तो ये चीज होती है हमारे पास। अब not only here, for example, I am a doctor, I am a teacher. If I would go to work at any of the or fries or ice cream together, yes. I know this one person who eats their fries with McDonald's की वो जो McFlurry होती है ना. And every time they do that, I give them the look. Recently, one potato and two potato came up with like a year or two ago, they came up with chocolate fries. Just thinking about chocolate over fries, it's disgusting. Yes, they did. Right. So now, so again, I I am expected to have a particular wardrobe. It would not look nice if I would go to my workplace in shorts and a t-shirt with flowers on, right? This would not, or that particular dress would not complement with my profession. So complementary things are those that go together. Same goes for the enzymes and the substrates. Okay, they need to be together. They need to be complementary to each other, whether it's in the shape or whether it's a chemical nature. एक आसान से एग्जांपल लेता है ना कि भाई अगर मेरे पास एंजाइम for example if the enzyme was positively charged, the substrate should be negatively charged, and vice versa. If the enzyme does not have a charge, the substrate should also not have a charge. So it's stuff like that. तो देखो, बात हमें क्या समझनी है कि देखो enzymes are specific for the particular substrate, पहली बात। अच्छा, दूसरी बात, अच्छा अब हिबा तुम्हारा वाला सवाल कि sir, once the products are formed, why do they leave the active site? Because the products and the enzymes are not complementary to each other, right? So what happens is, once a substrate is converted into the products, the products leave the active site, and the active site becomes empty once again. And the active site, it becomes empty once again. Now let's say if there were other substrate molecules of the same type, can a new substrate molecule of the same type bind with the active site of the very same enzyme? For example, let's put it this way. This enzyme that we have drawn here is amylase, and this was starch. Now let's say in a mixture you only had one molecule of amylase, one amylase, but you had four molecules of starch. Out of those four molecules, the first molecule of starch went to the onto the enzyme and was converted into its products, leaving behind three more molecules of starch. Can
Um,
another starch molecule bind onto the active site once again? हाँ जी बच्चे बोलो.
Yes.
Yes it can. Yes it can. Same है ना? वो हमारे पास substrate ही same है. So this makes enzymes reusable. Now there is a very popular question that usually comes in our exams that explain or suggest for 2 marks why a small amount of enzyme is needed for a large amount of substrates, right? So we would tell them that since once the substrate binds onto the active side of the enzyme, it's converted into products which leave the active sites, allowing the active site to be empty once again, so that the other molecules of substrates of the same type, obviously, the other molecules of the or other substrates complementary to the active site can bind onto it, right? Now, keeping these 2 points in view, we came up with a hypothesis, and that hypothesis is called as the lock and key hypothesis, right? Acha dekho, we have locks, right? So a specific lock has a specific key, which again is complementary to it, which could fit inside of it. Now that lock cannot be opened by any other key, master key is aside. Okay? Now let's say if that lock already has a key inserted into it, the keys can have duplicates, right? I can have duplicate of I have 2 keys for my car, right? I can, let's say I make 4 of them. So if one key is already inserted into the lock, can I insert a second key? नहीं, जगह ही नहीं बचेगी. हिब्बा reusable वाला मैं बता रहा हूँ. Give me a minute. So to insert a second key of the same type, I would have to take the first key out. इसी तरीके से वही reusable वाली बात कि भाई why are enzymes reusable is because once the first substrate would bind onto them, at the very same time कोई दूसरा substrate again complementary to it cannot bind to it. Only when the substrate is converted into products and those products leave the active site and the active site becomes empty once again, एक दूसरा substrate आके उसके ऊपर लगेगा. Again, complementary to the very same enzyme. This is what we mean by being reusable. कि one enzyme can catalyze many substrates provided that the first substrate should be converted into products and the active site of that enzyme should be empty. बात समझ में आई बेटे?
Sir, why all those substrates cannot like bind up together?
जगह ही नहीं है ना लड़की. For example, let's say in a cinema you have a seating capacity of 100 people and all the chairs are full. Can you go and sit on on one of the chairs? No, obviously the cinema people would not let you in the first place. Yeah, I common sense वाली बात ना. अगर मेरे पास, let's say, if I have up some water in a cup already, if the cup is filled up to the brim, can I add in more water? No, I can't. There is no space. समझ आई लड़की
Yes,
बात?
sir. Yes,
अब
sir.
देखो अब देखो वही वाली बात ना. अगर भाई फॉर एग्जांपल भाई ये वाला एंजाइम अपनी एक्टिव साइट पे जाके लग गया, अब एक्टिव साइट में जगह ही नहीं है ना, तो दूसरा वाला सबस्ट्रेट कैसे आके लगेगा? ठीक है. अच्छा, तो भाई ये हमारे पास लॉक इन की हाइपोथेसिस आता है. इसमें बात क्या याद रखनी है कि लॉक क्या है, की क्या है. अच्छा, क्या चीज किसमें insert, देखो key is inserted into the lock, right? Similarly, the substrate is inserted onto the active site of the enzyme. So where the enzyme is the lock while the substrate is the key. Right? यहाँ तक बात समझ में आ रही है? यहाँ तक कोई मसला? All right. ठीक है. अच्छा, अब यूजुअली एग्जाम के अंदर एक सवाल आता है कि explain the mode of action of enzyme using the lock-in key hypothesis, right? So we're going to write this down. तो हम लोगों ने कहा कि भाई the question is, so this चलो ये लिखता हूँ. Explain the mechanism या फिर mode of action of enzymes using lock and key hypothesis, right? तो भाई हम लोगों ने कहा, अब देखो मोड ऑफ एक्शन तो पूरा बता रहा है ना. So we said that for an enzyme to, or for an enzyme to catalyze a reaction, it is important that the substrate should have a complementary shape to that of the to the active site of the enzyme, right? So just let me read this out for you. For an enzyme catalyzed reaction to take place, it is important for the substrate to have a complementary shape to the active site. Once attached onto the active site, the activation energy is decreased and the substrates are converted into the products. उसके बाद हम लिख दिया since the ये तो mode of action हो गया ना, using lock and key hypothesis. Since the substrates are specific, or rather are specific बजाय हम कह देंगे have a specific shape to the active site, and bind to them, or bind to it rather, they are considered to be the key, while the enzyme acts as a lock. यूजुअली बच्चे ये तीन अगर हम मोड ऑफ एक्शन वाला तकरीबन 3 मार्क का आता है, 2 मार्क्स हम लोग इसके ऐड कर देते हैं, तो ये टोटल ऑफ अबाउट 5 मार्क्स अगर हम लोग उसको यहाँ देखें तो, राइट.